Hi guys, welcome to this video where I'm going to be covering the entire topic of OCR A-level immunity, all to do with those three big hard areas that we never quite understand. We've got our phagocytosis, and we've got our T lymphocytes and our B lymphocytes. So if you watch this video, hopefully you'll get your head around it and you'll be able to answer all of those hard exam questions. So guys, before we get started, make sure you click the link in the description to access a load of free flash revision cards, all based on this topic. And if you put your details in, I'll send them over to you immediately. So without further ado, let's get straight into that video.
Right guys, so we're going to try and get our heads around the immune response, which is quite a difficult topic because there's lots of different stages to it, and there's also lots of detail to it that you need to know about. For OCR, there's also a few little add-on things that you need to know about that you don't need to know about for AQA, A-level biology. So in this video, I'm going to focus on OCR content, more or less the same as AQA, but with a few little extra bits, and I'll mention them as we go through them in case you are watching this as an AQA A-level biologist. Now, I just want to make sure you know that there are two different types of immune responses, or two stages really to our immune response. We have our non-specific stage and we have our specific stage. We're actually going to start with the non-specific stage, but I'm not going into the details of kind of the barrier mechanisms because that's kind of the stuff that's a bit more simple. If you do want me to do a video on that, then make sure to comment below the video after you've watched it, and I'm happy to do an extra video on those barrier mechanisms. But just as a very quick overview, we do have some barrier mechanisms which prevent entry of microbes, keep the pathogens out. That might be things like having acid in our stomachs, or the fact that we have enzymes in our tears, we have wax in our ears. There's all sorts of different ways that our body prevents microbes from entering. And even if we cut ourselves, we go into our blood clotting mode and we go into wound repair. We also have inflammatory responses, which means that areas that are subject to possible infection, you will get swelling in those areas, which is brought about by a chemical called histamine, and that attracts kind of phagocytes to that area and begins a more specific immune response after phagocytosis. But as I said, I'm not going to go into all the detail of that in this video. This video is about the really hard stuff that we tend to get a little bit confused about. We're also going to talk about the details of the specific immune response involving the T and the B lymphocytes, which are very specific, special types of white blood cells that we need. Let's crack on with it. Starting with fever then, just a few little add-ons. So this, for example, is an OCR add-on. Why is it that we get fever when we are ill? Now, our normal body temperature is set at around 37 degrees C, and that's very much homeostatically balanced by actions between the hypothalamus and our cooling down and warming up mechanisms. Very important to maintain that optimum temperature at which our enzymes work best. However, when we are infected by a microbe, purposefully, our body temperature rises slightly, and that's because we want two things to happen as a consequence of that. By the hypothalamus resetting your body temperature to slightly higher than it should be, not to the point it would kill you, but just slightly higher, it in fact means that most pathogens are not going to be able to reproduce as effectively because they reproduce at their best at normal body temperature. So raising the body temperature slightly inhibits their reproduction, and at the same time, higher temperatures, slightly higher temperatures of our body enable our immune response to work at a bit of a faster rate. The reason why the hypothalamus knows to increase your body temperature slightly is because of chemicals that we're going to talk about today called the cytokines. Chemicals are like proteins that come from cells that are infected or damaged, and they're chemical signals, and they signal to other parts of the body and other cells that something is wrong. So in this case, a pathogen invading causes infected cells to become it causes the infected cells to release these cytokines into the blood, which ultimately stimulate our hypothalamus, and the hypothalamus resets the body temperature and makes it go up slightly in order for these two things to happen. Now, the first major part of our immune response, once we have actual invasion, so if microbes have got past all of our barrier mechanisms, is the process of phagocytosis. Now, phagocytes are a particular type of white blood cell. They come in slightly different forms. So you might have heard of macrophages or neutrophils. They're both types of phagocytes. They essentially do the same thing. They're just slightly different in shape and size, and also in their complexity and how they carry out the phagocytosis process. Right, so where does it all begin? Now, the first part of phagocytosis is the phagocyte has to be attracted to the pathogen or attracted to cells that are invaded by pathogens. I'm talking viral infected cells here. And in doing that, by being attracted towards pathogens or pathogen infected cells, they carry out a process that we call engulfing and digesting. And we're going to look at the details of that in a second. Now, this is non-specific, guys. It's not a specific immune response because it doesn't matter what the pathogen is, regardless of the pathogen type, phagocytosis will happen as long as an antigen that is foreign to our body is present in our bodies. Let's talk about recognition and attraction then. So what attracts the phagocyte? The phagocyte detects pathogens because of their foreign antigens on their surface. Antigens are proteins on the surface of cells. We all have them on all our cells. When we have our own antigens, our self-antigens, our body knows that those cells belong to us. It's self-recognition. So usually our immune system won't attack those cells unless you have an autoimmune disease, but that's another lesson in itself. Now, the phagocyte will detect anything that has a foreign antigen on its surface, typically a pathogen, or a cell that's presenting antigens from a pathogen. We also get chemical attractions called chemotaxis. The pathogen will release chemicals, or damaged cells, that means cells that have been invaded by a virus, will release chemicals, and that also helps to attract the phagocyte. Now, there are names for these particular chemicals. These are called the cytokines. Cytokines, therefore, are chemicals released by pathogens or by damaged cells, which help phagocytes to locate those cells in order to carry out phagocytosis. Cytokines, just in a nutshell, are proteins. We describe them as protein messengers. They carry out cell signaling, so they signal from one cell to another. And as I said before, usually these cytokines are coming from pathogens themselves or damaged cells at the site of wounds or ones that have already been invaded by a virus. So, neutrophils, what are these? Now, neutrophils are a type of phagocyte. They are known to be the first on the scene. So they're like your emergency services. They are first to respond to a pathogen that has entered your body. They will move towards the wound, the damaged cells, because of these cytokines, these chemicals that are being released by the damaged cells. And they also encourage more phagocytes to come to that area. Now, the phagocytes will now know that the body is under attack because of these cytokine chemicals coming from the damaged cells. But the phagocytes, lots of phagocytes, will start to migrate, move to those areas of infection and areas of inflammation. Don't forget as well that the cytokines signal to the hypothalamus. They're chemicals in the blood. So when they reach the hypothalamus will then raise the body temperature slightly. That will help to stimulate the white blood cells to work faster, and it will also reduce the rate at which the pathogen is reproducing. What happens during phagocytosis then? Once the phagocyte approaches the pathogen or the infected cell, which will have antigens on its surface, usually from the pathogen, or it will be releasing the cytokines, then the next stage is called engulfing. Now, engulfing is when the phagocyte literally extends its membrane. You can see it in the diagram here. It extends its membrane. They're quite flexible, so that it literally kind of molds around the pathogen and they bind together. So you can see that you've got receptors here on the phagocyte's cell surface membrane, which will bind to the actual pathogen itself. On doing that, the phagocyte will engulf, that means takes in the pathogen and encloses it into a little vesicle, which is a membrane-bound sac that we call the phagosome. You can see that in here, this is our phagosome with the pathogen inside. Once the pathogen has been enclosed into the phagosome, that's when you're going to get the next stage of phagocytosis. We call this digestion. You can describe it as breaking it down as well. And this involves another type of membrane-bound sac, little vesicles called the lysosomes. Lysosomes are organelles that we find in our cells, and what they contain are digestive enzymes, hydrolytic enzymes if you like, or if you want to be very specific, we can call these the lysozyme enzymes. Now, once the lysosomes fuse with the phagosome, we say that it forms a phagolysosome. Bit of a mouthful. Lysosome plus phagosome equals a phagolysosome. So basically it's these two merged vesicles together, but in doing that merging, it means that the enzymes in the lysosome can then be released into the phagosome and start to digest the pathogen or break it down. Once the pathogen has been broken down, and you can see in my diagram here that inside this phagosome, you've got all this broken down debris of what's left of the pathogen. But what the phagocyte then does with the antigens, the foreign antigens from the pathogen's own cell surface membrane, is it embeds them into its own membrane and presents them. It's literally like presenting them to the other immune system cells to say, look, this is what we need to target. And we call this antigen presentation. And we would describe the phagocyte in this state as an antigen presenting cell. Now, this is so important because if you don't antigen present, then the other white blood cells, which are part of the more specific immune response, won't have a clue what's going on. So it's a way of communicating to other cells that need to get involved, that they need to come to the rescue. And the T helper cells are the first on the scene. So what antigen presentation is doing is it's activating the next stage of the immune response, which is specific because the T helper cells are responding to a particular antigen, a specific antigen that's being presented on the surface of the phagocyte. Now, a few little details to add in here, and this is really just for those OCR notes, guys. So if you're an OCR A-level biologist, these are little things you might come across in like a multiple choice question, for example. You might come across this term major histocompatibility complex. Again, another mouthful. But what actually is this? Now, macrophages, like neutrophils, are also phagocytes, but when they carry out phagocytosis, it's just a little bit more complex, the process that goes on inside the phagocyte. What happens is when the macrophage engulfs and digests the pathogen, just like the neutrophil will do, it causes things to happen inside that are a little bit more complicated. It combines the antigens of the pathogen surface with its own special glycoproteins that are in the cytoplasm of the macrophage, and we call that kind of binding, the combination of them together, the MHC, or the major histocompatibility complex. Just a little add-on you need to be aware of. But ultimately, it's still antigen presents and it's still activating the next level of the immune response. First involved in that, in the specific immune response, are our T helper cells. Other little add-on to mention, guys, the opsonins. Sometimes you see this in a question as well, but what actually is it? Opsonins are antibodies, proteins, and what they do is they're found naturally in our blood. They bind to foreign antigens and they make the pathogen more obvious to the phagocytes. They allow the phagocyte to recognize the pathogen more clearly. We sometimes therefore say that the opsonin is tagging the pathogen, making it clearer to the phagocyte and encouraging phagocytes to migrate towards that area where the pathogens are and allow phagocytosis to happen more effectively. That is the kind of end of the phagocytosis bit. So what we're going to move on to next is what then happens with these T helper cells which have been activated by the phagocytes. Okay, so what are these T lymphocytes? We have already in our bodies, when we're born and as we kind of, our immune system matures, we have a library of different T lymphocytes. That means that we have, you can see here in my picture, a T lymphocyte, but we have a multitude of different types, which means the receptors on their surface vary. So you have billions, oh, I don't know if it's billions, but you have a lot of T lymphocytes of a variety of different types with different receptor shapes on their surface. And that's important, so that when an antigen is in our body that's foreign, there will be a T lymphocyte somewhere within our library of T lymphocytes that happens to have a complementary shaped receptor that will bind to the antigen that's being presented, for example, by a phagocyte that we've just been talking about. Okay, so let's see how it all works. Now, we call this the cellular response. So the T lymphocytes basically are cells that respond to cells, antigen presenting cells, and give rise to more cells. So it's all about cells. What they then do is they carry out a particular type of attack, and they also give us immunological memory. So let's break this down a little bit to make it a bit more simple. Like I said, we call this the cellular response. The reason they're called T lymphocytes is because although all your white blood cells are made in your bone marrow, where they end up maturing differs, and T lymphocytes made in bone marrow end up maturing in an area around your neck area of your body called the thymus gland. So T is kind of thymus linked, T lymphocytes. Now, their job is to respond in a specific way, because what they're doing is they're responding to the antigen, the specific antigen that's being presented by antigen presenting cells. T lymphocytes respond to antigen presenting cells. Now, we've been talking about in this video that they're phagocytes, but in fact, there are other cells that can be antigen presenting. Cancerous cells, so when your cell division goes wrong and cells become potentially cancerous, their antigens will be weird, they'll be foreign, and the T lymphocytes are responsible for recognizing that and responding and getting rid of cancer cells. And that happens all the time. It's only when that process no longer works properly that cancer can then obviously take over. Cells that you might receive in a transplant from someone else are likely to have slightly different antigens. They can also be detected by T lymphocytes as being foreign antigen presenting. So they would also be classified as antigen presenting cells. And cells that are infected with a virus will have antigens on their surface that are viral or a bit unusual, and again that would be recognized as foreign, so they are also classified as antigen presenting cells. So antigen presentation can come from a variety of different cells. We've majorly been talking about the phagocytes in this video so far. Now, just so you know what generally is going to happen here is our antigen presenting cells. Remember, this is usually a phagocyte, or it can be a cancer cell. It could be a transplant cell, or it could be a viral infected cell. Either way, you're going to have these antigens on the cell surface membrane of this cell, which are a specific shape, which are being presented. That is going to cause a particular type of T lymphocyte to bind to those antigens, but the T lymphocyte that gets involved is the type that has the right receptor on its surface, i.e. the shape that complements the antigens being presented. So as I said before, you have a multitude of different T lymphocytes, but it's only the one that happens to be able to bind to the antigen being presented that gets involved, which is why we call this a specific immune response. Now, the first T lymphocyte on scene is called the T helper cell. Let's just go back a little bit though. Pathogen invasion, we've talked about that. We know about antigen presentation, and that's all to do mostly with phagocytes, and therefore we call anything presenting a foreign antigen an antigen presenting cell. Oops, sorry, I've gone a bit too fast there. Now, I've also mentioned that this then activates a specific T lymphocyte, and we call the first ones involved the T helper cells. The T helper cells that have a specific shaped receptor on their surface are going to bind to that specific antigen being presented. Now, that binding, that straight away binding between the T helper cell and the antigen on the antigen presenting cell is an activator. And we described this process initially as being called clonal selection, because this binding between the T helper cell and the antigen on the antigen presenting cell means that that particular T cell with that particular shaped receptor is being selected for cloning, i.e. it's being selected to then undergo very rapid mitotic divisions and produce many copies of that same specific T helper cell. Now, in doing that, as well as the cloning of that particular T helper cell, we also get something called expansion and differentiation. Now, expansion is the actual process of making multiple copies of that same selected T helper cell. And that's when the T helper cell, once activated, undergoes mitotic divisions to form multitudes of it. We call that clonal expansion. But there is a little bit of differentiation that goes on here, because you don't just get more of the T helper cells, you will get more T helper cells of that particular type, which we'll talk about in a second, but we also get a slight differentiation process going on during clonal expansion, which leads to T memory cells, T killer cells, and T regulator cells. All of them have the same receptor type on their surface though, the one that was selected for initially in clonal selection. We're going to have a quick look at what all of these different T cells do in this particular immune response. Now, the T helper cells, once they're activated and they are clonally expanding, so you're making loads of them, they actually are then responsible for informing a particular B lymphocytes. That's where the B lymphocytes get involved. So without T helper cells, B cells won't get involved either. That's the next section of the video though, so we'll talk about that in a bit. In the meantime, we've got these T memory cells, vitally important, because what they do is they're the ones that give us long-term immunity. What will happen with them is if you encounter the same pathogen again, you will get what we call the secondary immune response, which is a very, very quick response to the pathogen, and it helps you to respond in a much more exaggerated way. Again, we'll talk about that more in a bit. The T killer cells, now these are really, really important, because what they literally do is kill infected cells. Those cells that have got viruses inside are quickly killed by the T killer cells. But we also have the T regulator cells, which are involved with suppression of the immune system. Might seem a bit odd, but it's something that we do need to do. We're going to look at each one individually. Now, the T helper cells, as I said before, we're going to talk about them a little bit more so later. In terms of the T killer cells, how do these clonally selected T killer cells work? What they do is they release a chemical called perforin. A perforation means to make holes in. Perforin is a chemical that creates pores or holes in the membrane of any cell that's been infected by the virus, for example. Now that means that that cell becomes fully permeable, so it increases the permeability of that particular cell that the perforins had action on, so it makes it no longer able to control what moves in and out of it. Literally, that means the cell is going to fill with fluid very quickly, swell up and burst, and that anything inside the cell is therefore going to be prevented from replicating. The virus can't replicate without a host. You've killed the host cell, the virus can't replicate. Memory cells. So what do these do? The T memory cells of the specific one that's been selected and cloned becomes part of your immunological memory. They stay in your blood literally forever, if not a very, very, very long time. And if they encounter that same antigen again being presented, they go into clonal expansion really, really, really quick. And they start to form, because they differentiate as well, into tons and tons and tons of that specific T killer cell. They clone into them again, and they will then destroy the pathogen really quickly and prevent it from causing any kind of symptoms. So you won't even know you've encountered the same pathogen again. Your immune system will deal with it super fast. Action of the T regulatory cells. Now, the regulator cells are really vital, because you always need to stop the immune response once it's done its job. It controls and regulates your immune system. Once the pathogen has been eliminated, you don't want to carry on with that immune response. It would be pointless, it wouldn't be efficient energy-wise, so it needs to come to a halt at some point. Also, we have special T regulator cells which recognize body cells that are yours, that have your self-antigens on their surface, and prevent your white blood cells from responding to them in an immunological way. So it ensures we don't have autoimmune responses. Sometimes we can get autoimmune responses where things do go wrong with your regulatory mechanisms. And also involved are chemicals called interleukins. Inter means between and leukins means white. So interleukins are actually another type of cytokine, which I'm going to mention in a minute, which are specifically chemical signals or signalers between different types of white blood cells. T helper cells, let's just go back to them a minute. Remember the T helper cells, which are first to get involved in the specific immune response. When they bind to the antigens on the antigen presenting cells, they produce these interleukins, which are these cytokines which communicate to B cells, the B lymphocytes, and attract them towards the area, and also stimulate them into their own kind of clonal expansion. Again, that's our next section of the video. So for this bit, I just want to say that the T helper cells, on binding to antigens on the antigen presenting cells, produce cytokines, which are specifically interleukins, chemicals between white blood cells, activating B lymphocytes to get involved, and also stimulating more of those phagocytes, the macrophages, allowing them to carry on destroying pathogens and foreign cells, not foreign cells, and infected cells, so that they don't continue using your own body cells to reproduce. So you're going to make sure that the T helper cells keep the process going for as long as it's possibly needed. So why is this cellular response so important? Cells responding to cells and cells killing cells means that you're going to target viruses really effectively. Remember, these are inside your cells. They are inside your body cells. We call them they're almost parasitic, aren't they, I suppose, because what they're doing is they're getting inside your own body cells and using your own body cells machinery, organelles, to manufacture more viruses. So we want to kill any viral infected cells quickly to stop viruses having the opportunity to reproduce. We also want to make sure that cells that could become cancer are destroyed quickly. And we want to make sure we have memory of that particular antigen by creating memory T cells during the cellular response. And don't forget that the T helper cells, in the production of the interleukins, which are a type of cytokine, activate the right specific B cell or B lymphocyte to also become part of the immune response, which is the next stage of our video. T helper cells also release other cytokines or chemical signals which stimulate the phagocytes to carry on doing their job, to ensure that you are killing any cells which are invaded by pathogens, by the viruses, as quickly as possible. We're going to move into our very last section now on the immune response and look at how the last type of white blood cell gets involved. It's still a specific response in this case, just like with the T cells. Now we're going to look at what we call the B cells now and how they get involved. The B cells, just to put it into the context of why we call them the B cells, like the T cells, they are also made by the bone marrow, but they stay in the bone marrow and they mature in the bone marrow, which is why we call them the B cells. We also describe this as the humoral response because B cells give rise to antibody proteins which are soluble that end up in the fluids of our body, and the fluid of your body is called humor. The humoral response therefore involves body fluids and antibodies, soluble proteins in the fluid of your body, of your blood. Humoral response is specifically to do with B cells and the production of antibodies. Now it says here by plasma cells because B cells give rise to particular cell types called plasma cells that release antibodies into the humor of the body, the fluids of the body. Still a very specific immune response, because the B cell that gets involved in an immune response will be directly involved with a specific antigen being presented. So like with the T cells, the particular B cell that gets activated and gets involved in this response will be to do with which particular antigen being presented. We also, like with the T cells, get memory of the antigen and B cells that become memory B cells for life, so that if you encounter the same antigen again, you're going to respond super quick. So we're going to look at how all of this works. The stages of the humoral response. The first thing that has to happen is the right B cell has to be selected. That means that the B cell with the receptors on its surface that complement the antigen being presented have to be selected first. And just like with the T cells, it's when B cells with a particular shaped receptor on its surface bind to an antigen of complementary shape that's being presented. And that activates the B cell. Now, the T cells also play a big part in this activation process. So let's have a look at how this works. The receptors on the surface of the B cells are actually, more precisely, specific antibodies. They're proteins on the surface of the B cell, which are antibodies. They're these little Y-shaped proteins. And you've got loads of different B cells in your library with different antibodies on their surface of a particular shape, of different shapes, that will recognize and bind to complementary antigens that are being presented when you've been invaded by a pathogen. When the B cell receptor, that's the antibodies on its surface, bind to a complementary antigen, the B cell also weirdly takes in the antigen, kind of processes it, and also presents it on its surface. So what happens is you get your B cell, so this is your B cell, there's its antibodies, there its receptors, it's encountered foreign antigens, engulfed it almost, it's not phagocytosis, we just say it kind of processes foreign antigens and presents them on its surface because they are bound to the complementary antibody. So we've got the antigens, that's my little circles here, and the antibodies, which are bound together and being presented. When they present those antigens, remember the T helper cells, those specific T helper cells, which are also involved in the same immune response because it's all to do with a particular antigen, they will then start to bind to those antigens as well. So here's your T cell. They're the ones that were clonally selected and expanded during this same immune response. They will have receptors on their surface which also bind to those antigens now on the B cell's surface on their antibodies. Then what happens is the T cell releases cytokines called interleukins, and those chemicals signal between the two. And what those interleukins do is it makes that particular B cell very, very active. It activates it. But what does that actually mean? When the B cell gets activated, it goes into its own clonal expansion. So that particular B cell with those specific antibodies on its surface go into very, very rapid mitosis, or we call it B cell proliferation, which means very, very, very fast division. These B cells don't just proliferate or clone. Like with the T cells, they also undergo a little bit of differentiation. Now, just to remind you that when we get these fast mitotic divisions making loads and loads and loads of the same, in this case, B lymphocyte, we can also call this clonal expansion. You're cloning one particular cell type hugely, so you get tons and tons of that same particular B cell. Now, when it does undergo a little bit of differentiation, you then end up with slightly different forms of that same B lymphocyte. The plasma version, the plasma cells, now what they do is they start to release large amounts of that antibody. The antibody that was on the surface, they release loads of free antibody of the same type into the humour of the body, into the fluids of the body. B effector cells are formed as well, and they divide and form more and more and more of these plasma cells. So they keep making more plasma cells, which ultimately lead to more and more antibody in the humour of your body. We also get B memory cells of that particular B cell and that particular antibody. Just like the T lymphocyte, they will literally stay in your body fluids, your humour, your blood for a very long time, possibly life, and will respond quickly and undergo clonal expansion and antibody production really, really super fast and exaggerated if the same pathogen, the same foreign antigen is encountered at a later date. So what we have now is lots and lots of free soluble antibody of that particular one that's been selected from the B cells that were activated. We have loads of this antibody now in the fluid of our body, in the body humour, in the blood, of the plasma of our blood. But what do antibodies generally do? Antibodies, which are these Y-shaped proteins. Now, again, if you do want a video on antibody structure, drop a note below the video and I can do that as well, because that's another big topic in itself. But what antibodies generally do is they carry out specific actions. So remember, these are coming from the plasma cells, the cloned B lymphocytes that are plasma cells, giving rise to a specific antibody into the body's fluids. And they basically work in the following ways. They cause agglutination, which clumps pathogens together, making the phagocytes have an easier job, because if all the pathogens are clumped together, the phagocytes can get to them easier and carry out phagocytosis. Opsonization, antibodies can coat the pathogen, making it easier for the phagocytes as well to carry out their process of phagocytosis. And opsonins, also remember, is about, we call it tagging the pathogen. So it makes the pathogen more obvious to the phagocytes, so they can have their action really, really quickly. And neutralization, antibodies can also bind to toxins that come from the pathogens, like viral proteins that end up in your blood and make you feel really ill. And they can also stop viruses from entering and infecting body cells. So they neutralize the process. And then there's just a few little diagrams here to show you what we mean by agglutination. You can see here that these pathogens have been clumped together by the action of the antibody. So the phagocyte literally can migrate to one area and have a big exaggerated effect. And here you can see that the antibodies, which are coating the pathogen, also prevent it from entering the host cell. So if this was a virus, if an antibody is coating the virus, it's not going to be able to invade the host cell and cause all those issues. Immunological memory then, also a big part of the B cell response. Memory B cells remain in your blood way after infection. As we mentioned before, if they encounter that same foreign antigen again, or the pathogen with that antigen on its surface, the memory B cells rapidly divide into those plasma cells, produce loads of those soluble antibodies into your body fluids, your body humour, which is your blood, and we end up with what we call the secondary immune response, a fast, strong response that you wouldn't even be aware of happening because you wouldn't get any symptoms because it's that quick. Right, so that kind of brings us to the end of our immune response. It is complicated, it is hard to get your head around. So watch a few times, and if you've got any questions, make sure to ask below the video in the comment boxes.
Guys, if you've enjoyed that video and you're after even more, then you can get hold of all our free resources, my resources on the My Edspace website, where you'll get revision notes on loads and loads of biology areas. You can also access videos, exam style questions and solutions. Make sure to check that out. And if you want even more help, then you can also sign up for one of our courses where you'll have access to 2 live lessons every week. You will also get workbooks to go with those lessons, homeworks and video solutions. It's all there. You just need to click the link in the description. And guys, if you really did find this useful, then don't forget to hit subscribe, drop a like and mention in the comments any other topics you'd like us to cover that you would find useful. And I'll see you in the next one.